computer. Recording, share screen. So, spirochetes, you have a look of one behind you here on the PowerPoint, okay? So, a spiral notebook is great. I see some have spiral notebooks still. They still make those. And then you see a big slash and you see Legionella out there by itself. And so that's what we're covering today. So we got about three emphasized spirochetes, and then we'll hit Legionella, which is not a spirochete. Okay, so spirochetes are helical shaped. They have no external flagella, but they are modal, so they can move. Okay, so they're kind of snake-like, and they do move around. And we have three genus or genera: Leptospira, Treponema, and Borrelia. All right, so we have, hopefully you may be not as familiar with lepto as you shouldn't be, but you should be very familiar, or you will become very familiar with Treponema and Borrelia. You definitely are familiar with. So we will see those in micro. And what happens here in this lecture, for those that aren't in immunology, okay, again, back of the room, immunology, we're going to cover the test. We're going to mention the test here in micro, but in immunology, we're actually going to do the test. So that's the connection between the two courses. So I'm not going to spend a lot of time in micro going over testing methods when we're going to get that in immunology and lab. Okay, so I hope you're not disappointed if I'm like, oh, he just mentioned that, but he didn't really explain it. So don't think it's the last time you're going to see Treponema or Borrelia. So Leptospira species, what you're going to see is that it is commonly encountered in human infections, so it's not like something we don't have or don't, never see. But it's leptospirosis, and you see that we have some subspecies down here. So we have Leptospira interogens, and we have a serovar of icterohemorrhagia, and that's from rats, okay, known as Weil's disease. We have serovar canicola from dogs, okay, and that gives humans infectious jaundice. And we have Pomona, which is from pigs and cows, and we get Swinehert's disease. Okay, so hopefully you're picking up on something. Anybody want to share what they're picking up on?
Animals.
Yeah, these are zoonotic infections. All right, if we're messing around with these zoonotics, they jump, not newly jump, not like COVID, but these can jump to human infections just that way. Leptospira has a where it's found, it can be found in the blood, it can be found in spinal fluid. So those two parts of our body, okay, and blood, connective tissues, those spinal fluid, those are supposed to be what? Sterile. All right, we're not supposed to have organisms circulating in our blood. We definitely don't want organisms in our spinal fluid. So keep that in mind. I don't know how much you've heard about cerebral spinal fluid collection and why we do that in healthcare, or blood cultures, why we do that in healthcare. But that is to make sure that the patient hasn't gotten the infection to go past where it normally would be and moved into the bloodstream, or even worse, moved into the spinal fluid. Okay, because that's when we get into trouble. This is when the patient doesn't do well, right? If we don't get that under control. So as you see, after the first week of infection, the Leptospira, that spirochete, enters the kidney, and they're shed in the urine. And we can do a dark field microscopy to find them in the blood, cerebral spinal fluid, or the urine. They end up after a couple of weeks there in all three, or could be. but they're rarely seen on direct examination because they're so small in number. So it doesn't take a lot of these spirochetes to really cause some problems. And that's what we're going to find a lot with spirochetes is, it's not like our staph, our strep, or our Neisseria where we can plate that out. Okay, we're not going to be able to plate out a spirochete and have it growing. So we got to do kind of a direct examination. Okay, so you see all this lit up with, you know, dark field fluorescence, or, you know, really looks, or we can stain, right? Stain the tissue that we're checking. So we have Gomori's methanamine silver stain. Cultivate easily. And we have, if we go to Leptospira media, we may be able to isolate it from the specimen itself. So I'm not too worried about Fletcher, Stewart's, Leptospira media. But here what you will find on direct examination of a stain. Okay. And you can definitely tell there's something weird about this tissue that we've picked up, or fluid we picked up and examined. So this is where you're, you know, if we were able to, optimum growth, if we were going to grow it, we were able to get it going 30 degrees Celsius. So we're doing what with Leptospira? Decreasing the temp. Observation, we look for tightly coiled spirochetes with hooked ends, and we identify it with a serological test. So what do we need to get from this is that we are going to take our sample, send it off, and do a serological test for ID to see if it's there. Nice pictures. That's electron microscopy there. Nice and good. Direct fluorescent antibody testing. Okay, so these are our immunoassay type deals where we light up an antibody that has fluorescence light to it, and it stays there and detects the spirochete. We actually can see the spirochete. So with lepto, we see that it is a zoonotic affecting many animals and species, including humans. And where we find it is rodents and domestic animals. So that's part of your fill in the blank from lecture. Transmission, urine from infected animals. So if the animal has Leptospira and you're exposed to its urine, then that's where how you would get infected. And infection is just contact with the urine, contaminated soil, water ponds, swamp, sewers, so, and food. So if you got, right, if you got a pig or a cow or a dog, right, with this, and they're peeing all over things, and then you are swimming or wading or eating food that's been contaminated with urine, then you could pick it up and be infected with it. So just through the mucosal membranes, it breaks in the skin. And then the diseases where we've started with this one, where the organs are infected, are central nervous system, kidneys, and liver. And then we have one that we want to associate with Leptospira, which is Wheels disease, the icteriohemorrhagia. Symptoms include jaundice, but what causes fatality or mortality in the patient is renal failure. All right, so that was lepto. Probably not going to see it, right? I've yet to be asked to try to identify Leptospira, but you never know. It might be one of those that we find every once in a while. But this one is definitely going to be one you're asked to do some screening on. And anybody know the Treponema that I'm referring to? Have you ever heard of Treponema before? Okay. All right, so a Treponema, again, a spirochete, just as Leptospira was. It is microaerobic, sorry, I was going to say aerophilic, but microaerobic, habitat, oral cavity, intestinal, so urogenital, mucosal. And so there it is. So you should be able to definitely tell when you have a spirochete picture. We have Treponema pallidum, subspecies pallidum. And we have some illnesses here that you need to know, okay, because the board, your exam that you take once you graduate, loves to mix in the different pallidums, right? The Treponema pallidums. We see one here, Treponema pallidum, venereal and congenital syphilis. So have you ever heard of syphilis? Anybody heard of syphilis? Okay, so when I said, have you heard of Treponema? Everybody said no. I said, have you heard of syphilis? Y'all said, yeah, heard of syphilis, right? So this is the Treponema pallidum. We have another pallidum, the pertinum, causes yawls. Okay, so we have yawls, and we have a non-venereal endemic syphilis. So the pallidums all call us some kind of syphilis. And yawls is one of those that, hey, it's a weird name. You don't see it in this country, but you see it in other parts of the world. And we'll go over that in a minute. And then you got keratium, penta. So yawls and penta and congenital syphilis, they like to put all those kind of together. And then they'll ask you, like, which one causes penta, which one causes yawls. And they'll still ask you a little bit about pallidum or syphilis, but all of a sudden you're like, well, we've never even, I remember that in lecture one day, we talked about yawls and penta, but I had no idea they were going to ask that on the board, and they will. Every once in a while. Say every time. But we have them associated with skin ulcers. We're going to see those. Periodontal gum disease and diarrheal illnesses. We have ways to go with human diseases with Treponema. Oops. So specimen collection for Treponema pallidum. Again, direct examination from the lesion or lymph node. We can do serological examination of the blood, the serum or the plasma. And y'all were all in principles, right? So just to check on principles. What's the difference between serum and plasma again? I know, this is the former phlebotomy teacher coming out every time. What's the difference between serum and plasma? Zoom, you can answer this one too.
Does it depend on which tube you get it from?
Depends on which tube, right? But there's a tube that does something to it, and there's a tube that doesn't allow it to do that something. So tell me which is which.
Well, the serum separating tube, I'm assuming that's serum that you get from it, right?
Yeah, but what did the blood do before you put it in the centrifuge?
It clotted.
All right, what's the other tube? So that's serum, and what's the other tube?
You
get plasma, or one of the ones you can get plasma from?
A blue top? I forget what it's called.
Plasma from blue, yeah. Or purple, or green, right? Are y'all familiar with tube colors?
Not the best.
Not the best. From principle?
That was the test that I didn't do so great on.
So the difference is what? Serum comes from separating of clotted blood fluid from the cells, and plasma comes from not not clotted or anticoagulated blood separation. Okay. They're both the liquid part of the blood. Just one comes from a different step, which is clot versus non-clot. That's the difference. So the question for new lab techs and phlebotomy, is always what? Are these different? Right? Are these different? Some of our tests will say we need serum, and some of them will say they need plasma, and that is a difference. So if you just don't know the difference and you're just like, well, that's just here, yeah, then you may be testing the wrong recommended fluid. So we definitely can't do blood clotting out of a red top tube. We can't measure the time it took, right, in a blue top like we do for a blue top and coag. All right. All right, so here, Treponema pallidum organism cannot be cultured in an artificial media. The culture is accomplished by inoculating, yes, I know this sounds awful, testicles of rabbits. Yes, we can take testicles of rabbits and we can culture out Treponema pallidum. That's something you probably didn't know before today.
How did they figure that out? That's what I was thinking.
Great mind, I guess, just trying something out. No, I don't know if the rabbit is alive. I don't know if the rabbit is dead. I don't know if they just took the testicles out of the rabbit. I don't know how that works, but I thought it was definitely something that we could go with. Again, we use dark field examination of the exudate or the lesion or the lymph node aspirate. So if the patient has a lesion, we can do a direct examination. And syphilis causes lesions. And we're going to see a lot of pictures of that here in a minute. And we can also do antibody testing. We can do antibody, and this is where we kind of move over into immunology, but we're not going to move all the way in. Does that make sense? So what we're going to do is hint at this, and then we're going to really cover it in immunology. So non-treponemal antibodies, yay, versus treponemal antibodies. And those, everybody in here is in immunology or had immunology. Supposed to know this, right? About an antibody. I shouldn't have to explain what an antibody is. But I have to, the specific antibody is what we're doing here with syphilis. And just like finding rabbit testicles grow this culture, we also found that there is a non-specific antibody is found in the syphilis patient, but there's other reasons that antibody could be formed too. But we actually have a test that we actually run to screen for this. And how many of you have ever had an RPR test done? Screen. You didn't have to have one before phlebotomy, or anybody at the hospital had to have one. We used to ask our students, since they were phlebotomists and they were doing a lot of contact with the patient, and we asked them to do an RPR screen for syphilis. Because if you have syphilis, you may be contagious with syphilis, so we always wanted to make sure that you weren't. Then we realized the clinical site really didn't care about syphilis, so we quit testing them for syphilis. We were just doing a test that I guess at one time one hospital decided we needed a syphilis screen. So I was just curious if anybody had an RPR. Usually we send them to the health department, and they do an RPR and an HIV at the same time. Okay, so you got a free HIV test with your RPR if you went to the health department to get it done. But what they did was they actually screened for a non-treponemal antibody. You guys are in immunology, and you're like, hey, Mr. Director, how in the world do we get a non-treponemal antibody with a syphilis patient? All right, shouldn't they have a treponemal antibody if they have syphilis? And that'd be a great question to ask, because that'd be true. If they do have syphilis, they should have a treponemal antibody. Okay? So this is the two things that we need to, we'll see with our tests that are about to pop up, which is, we got dark field examination. We see our spirochete. You should see that. It's obvious we have a spirochete. Obvious we have a spirochete. That silver stain again with pallidum. Obvious that we have a spirochete. Obvious we have a spirochete. But here are our tests. So I think to answer Mary's question is that how did they figure that out? Well, there's something called the Venereal Disease Research Laboratory. never heard of that either. But the first test that they developed was called the VDRL test, which stands for Venereal Disease Research Laboratory, where they're doing research on venereal disease. Okay, does that make sense? So they probably figured it out there. I don't know.
Well, I just don't.
All right, but the one we just mentioned was the RPR, which stands for Rapid Plasma Reagent Card Test. And we're going to do that in immunology lab. It's on our list of things we got to do. We're going to do that. Okay, but that's what they call the non-treponemal antibody. Okay, is the reagent antibody. So we'll do a test to detect that antibody. And what that is that we have these reagent antibodies that are called a Wassermann antibody, which are non-specific formed against the cardiolipin. Okay, and cardiolipin is part of the organism's cell. Okay, it's a phospholipid. Remember, phospholipids is part of the membrane. Remember that when we went back, we started in micro and we talked about the phospholipid bilayer. Okay, this phospholipid is called cardiolipin, and that's what the antibody is formed against. Okay. Well, I know you're like, oh, this is going to be really confusing. Specificity and sensitivity, these tests are sensitive, right? We can find it if it's there, but they're not specific. They don't tell us 100% that we got syphilis. They're a great screening test that lets us know there could be a syphilis case, but we have to go do the syphilis antibody after that to confirm it. So hopefully you're hearing a lot of that today in the last month about what test is good, what's this mean, is it specific, if it's missing, if it's false positives, false, all of that good stuff that you've heard for the last 6 months now, right? Is it 6 months already? You're now in those classes. Now you're in that way. We're going to start figuring these things out. So we're going to see that kind of terminology for all these other tests that we do. And this is one. So you'll see it as the VDRL/RPR test. It's looking, so when I give it to you on the test, you are looking for the non-treponemal antibody. It's an antibody formed not against the treponemal pallidum, the spirochete that we're talking about. What it's formed against is the cardiolipin, right? That may or may not be from syphilis. It may be from another organism. But it does detect if it's there. Okay? And we're going to see that. It clumps up in our little card. We did, we've done a card test once for staff. We're going to kind of do one here for antibody testing in immunology. Is there any questions on this? When I say sensitive but not specific. Everybody's got that?
So I have a question.
Yes.
So you said the non-treponemal. You kind of just said it, but I'm just making sure I'm hearing it right. So non-treponemal, the antibodies are non-treponemal antibodies, but they are antibodies against the cardiolipin, right?
Correct. They're not antibodies formed against treponemal pallidum. So here's the problem. We can get false positives, okay, because cardiolipin is found in many tissues and many conditions. Now, I know some of these conditions you're like, who's had malaria lately, right? Who's had scarlet fever lately? You could be going through scarlet fever, relapsing fever, we're going to see that. Or you can have chronic diseases like mono, TB. You could have lupus or rheumatic heart disease. All of those would, what? Produce a cardiolipin antibody. Okay, you'll have an antibody against the cardiolipin, which is known as a reagent. That's what this is. So we're going to test for the reagent. It's used for screening only. So if we get a positive RPR, then we have to go another step, which is to do a confirmatory treponemal antibody test to confirm. And we have that. I mean, it's not like we don't have that. We just don't want to use that for everybody that needs to be screened for syphilis. If we're like us, like in phlebotomy, we're just screening everybody to make sure you hadn't been exposed to syphilis in your life, which chances are probably haven't. But we could screen the whole group, the whole class for RPR and not have to use the treponema antibody unless one of y'all were positive. The treponema test is a specific antibody produced against treponema pallidum antigen. And we do have a way of doing that. One of them is the FTA, which is fluorescent treponema antibody absorption test, FTA-ABS. We have an immobilization test, a microgluten test, but the one I'm familiar with, the one you would lean toward doing would be the FTA-ABS. It's pretty easy to do. You just have a kit, open up the kit. You look for the antibody. If it's there, it's there. But so this would be to confirm infection. There's the FTA fluorescent treponema antibody absorption test. So what is clinically significant about treponema? If you look at it, this is the key. Syphilis is treatable, but this is where we have untreated syphilis. Okay, so patient is it's being exposed or got exposed. So, I mean, I'm not saying that increase your number of sex partners increases your chance of syphilis. Not being very picky about your sex partners could increase your chances of syphilis. Your living conditions could increase your chances of syphilis. Your health status, your economical status could increase your chances of syphilis. Okay, so what we see is that this patient, being knowing or not knowing, doesn't go get treated. Okay, so syphilis is all about not being treated. To go into the different stages of syphilis, and there's 4 stages of syphilis. The primary stage is this person knows they have something, they have a hard canker, okay, that contains the organism, is how it spreads. But if you don't get treated, it heals spontaneously. You lose the site of the canker goes away. It just so happened to be something I was worried about for a couple of weeks, and I'm back to my normal self again. Everything's good, right? That was actually a syphilis infection. If they had sex with anybody during that time, they infected that person or could infect them. So that goes away spontaneously, but you didn't treat it. Okay? The secondary stage is a rash. Rash covers the skin, the mucous membranes. And that rash also contains the organism, but it heals spontaneously. So you went from a canker, you had a sore in the genital area, and then it went away, and then you broke out in a rash. And if you're not worried about the canker the first time, you're not worried about rash. That doesn't look as bad as a canker. And then you move into secondary stage, and that goes away spontaneously. Well, what's happened is that you've set yourself up for a big chance of moving into these stages. And this stage is known as the latent stage, which we cannot, look here, the antibodies are not present, okay? Or the antibodies are, but the symptoms are not here. So if we tested the patient in latent phase, and we did a RPR, and we got a positive RPR, and we went and did the treponema antibody, they would test positive. Even in the latent stage, without the canker, without the rash. During this time, the patient is not contagious, and transmission is restricted to rare cases between the mother and the fetus, which would not be good, and this period may last for years, even for the rest of the patient's life. Okay? The good thing is we can find the antibody. okay, if we test them. We can find this patient in the late stage, even though they don't have the canker or they don't have the rash. Then, if we don't find that patient, then there's a small percentage of the patient that was positive at the beginning, untreated through the primary, secondary, and tertiary, the late phase, goes through the tertiary phase. This is known as late syphilis. And this is when we get granulomatous lesions, the guma. We'll see pictures in a minute. We have soft tissue affected, bone affected, central nervous system, and cardiovascular system. Okay? So the patient now is going into some deep problems, especially if it gets the central nervous system. Okay, so keep that in mind. So that's as far as I want to go today. We'll go back into syphilis when we get into immunology, and we'll go through a little more about the stages. But right now, the big thing is that you saw untreated, 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 okay? And then we know there's a percentage that doesn't go all the way into tertiary, okay? And we can test them in latent and find it. So here's the hard canker. So this is a sore looking thing, okay, that is contagious. So there is spirochetes right here, Treponema pallidum, spirochetes in that lesion. More lesions. More lesions. More lesions. It's a little harder to see that, but I think I know what I'm looking at. I'm not real sure there. More lesions. More lesions. Yeah. More lesions. Okay. I know. I'm going to have y'all not, yeah. Skin rash, secondary stage. Hands. So again, this is the rash that appears after the canker goes away. This is a rash that pops up, the untreated, secondary. Untreated, untreated. Then if that goes to latent, right? Latent is no symptoms, no anything, right? They're testing for the antibody, but there's no symptoms to them. And if they move into tertiary, right? Tertiary stage 4, basically. This is the guma. The guma on a nose. Guma on an arm, leg, maybe. Looks like lungs. Aortic aneurysm rupture. Neurosyphilis, demyelination, not good. And then they have congenital issues. This is the baby. We were talking about the baby being born during that transfer stage, that the baby could end up with syphilis. Causing birth defects there. Got a hole in the palate. Teeth are spread out. Hutchinson's teeth. Blindness. And this just gives you an idea in the late 90s that the rate in the United States per 100 births, and what do you think happened here? What do you think, how we got that down? How do you think in 1992 we went from per 100 live births is the primary, secondary, and congenital down to 98, we're down like under 20 live per. What do you think happened?
Did the screening test come out?
What's that?
Did the screening test come out then?
Yeah, we started screening for pre-OB screening. All right. If you're pregnant, you got an RPR screen. Got treated. So there was this non-congenital syphilis transfer. You got treated. Syphilis is treatable. Okay. It's not like, oh, we can't do anything. And that's why I wanted to emphasize that untreated, untreated, untreated you saw at the beginning, all those different stages. That's how it rolls through. If it's treated in the first canker, we're done. Okay. We treated it. We're not going into secondary or tertiary or in the neuro. We're not moving that way, okay? Keep that in mind. Story will continue in immunology. All right, so we had another one. We had pallidum, but we had a subspecies of Pertinum. And this is Yawls, so you need to associate Pertinum with Yawls. Distribution, tropical countries. Rural, can you say rural, right? Rural areas of tropical countries. The lesions affect the skin, soft tissue, and bone, and it's direct contact, person to person. So Treponema likes to transfer from me to you, all right? So that's how transfer happens. So it's skin touching skin, and we have transfer. So that's about it for y'all. So we'll look at some pictures, tropical area, skin lesions, bone being affected. Okay. So that was Yawls. So the big thing there is to understand which one's Yawls, which one pallidum is connected. And then we have Treponema caritium is penta. So penta goes with caritium. And we see it in the Central and South America. Skin lesions again, but we get this deep pigmentation of the skin lesion. Direct contact, person to person. This deep pigmentation. Again, look at the hands, body, back. There is our spirochete again. And then we have the last one is endemicum. Endemicum is an endemic syphilis, so this is not a sexually transmitted. It's more of a childhood disease. It's a non-venereal, so that means non-transmitted, sexually transmitted. Venereal is transmitted sexually. Highly contagious skin lesions, again, direct contact. And that's it, okay? All right. So we had what, four of them? We had Yawls, penta, and then a non-venereal syphilis. Y'all did good. You did good. Usually when I get to that antibody testing, you understood it, right? So the biggest thing is that antibody is to cardiolipin. It's formed during the infection of syphilis, but it's not specific to syphilis. It's a great screen. It has to be confirmed, and then once confirmed, we'll know the treatment. Okay. So now we move on to Borrelia. Spirochete, of course. And we have separate ones. We have one known as relapsing fever. And of course, it's just as the name sounds. This is going to be one where relapsing fever would be, we have fever, we get over the fever. A week later, we have fever again. We get over the fever, and a week later, we have fever. Okay. So this is a spirochete. It's transmitted by arthropod vector. When we have an epidemic relapsing fever, and we have an endemic relapsing fever. So epidemic is a louse-borne, human louse, body lice, transmitted by Pediculus humananus. That's the epidemic. So a great test question is relapsing fever, epidemic relapsing fever is transferred by what vector? And that would be lice, body louse, okay? The endemic is a tick-borne, but it's a soft-bodied tick, which doesn't look like the ticks that you're used to seeing, but we'll see pictures of the Ornithodorius species of tick. And how do we, blood collected during the febrile period would be how we would see this, okay? So we would detect it with the fever, and this was where this would come in handy when we do blood cultures, right? So we're always leaning toward blood cultures to be drawn when the patient is actually spiking a fever. When their fever goes up, hey, we're at 99 today, but we're at 102, we need to get blood cultures. I worked with an old-time doc when I first started, and the old-time doc said, every time my patient goes above 101, I want blood cultures. Okay, we had some patients, now it wasn't relapsing fever, but we had some patients that, once the Tylenol or Motrin wore off, their fever, they were still, it would go back up, and we had to draw blood cultures. So some patients had maybe 20 sets of blood cultures during their stay in the hospital. They kind of got tired of seeing me come down and draw blood cultures every time their fever went up. That was his way. He wanted that. He thought that was the key to catching something, was to make sure that when their fever went back up, to get a blood culture. So here's a picture of a body lass, if you've never seen one. Got the knit over here. You probably can't see that one. That's the immature stage. That's the one, you pull up the hair and you see little white eggs, and then you see the knit, and then you see the big, yeah. So this was, if you came through the lab, like when you were wanting to be a CLS student, we might have had this on the slide for you to look at. This is the soft body tick. So that was the epidemic. This is the endemic, right? Relapsing fever. This is the Ornithodorsus hermesii, soft body tick, which we're used to seeing the deep woods tick that you get when you're out hiking and camping. All right, questions? Borrelia species, we do direct examination again. We do dark field examination. We can do a drop of diluted blood, or we can stain it with our gizma or our right. Right stain is what y'all use for. Right stain is used for blood smear staining. Yes, there you go. All right. Growth requires what's known as a Kelly's medium, which is a liquid medium. It actually has stuff in it to help detect the spirochete. We don't do biochemical or serological tests, so we have to do identification based on the arthropod vector and the geographic distribution. Ooh, is that going to be tough, right? How would you like to be diagnosed because of which tick is found on you, okay, and where you live? Okay, and we're going to see that. We're going to see a very famous Borrelia here in a minute, that's how it got its name, because of its geographical location. You might want to take a shot. You might got to guess. I just like saying the name. I don't know, but here's the blood smear. Did a right stain, or if that's the gizma stain, I don't know which one, but you see the spirochete inside the blood, so direct examination. The symptoms resemble other febrile illness, so this relapsing fever, we get febrile episode, then after antibody response, the numbers decrease, so the afebrile period begins, and then we get a new antigenic variant, allows it to multiply again, we get fever again, we get an antibody response, it goes back down. So we have one that's called Borrelia recurrentis, which is the louse-borne relapsing fever, is only in humans. And we have a tick-borne, these are our reservoirs. We have a tick-borne relapsing fever, which is zoonotic, meaning rodents and rabbits are the reservoir and the source of human disease. All right, this is where I was trying to get you to, which was Borrelia. Who wants to go with the name? Who wants to try? Who's my English/minor major? English minor. Who wants to be at graduation and pronounce everybody's name when they walk across the stage? Who's that person? Who's my linguistic person? All right. So it's not hard. I just go with how it looks, right? I love saying it. I think I did a paper on it one time. Back in high school. You gotta do papers in high school. When you were in high school, I had to find some organism. This is the one I ended up. I just found like a journal, and I found this. Oh, this is new, right? It was Lyme disease. Borrelia. No, I can't. I've got Borrelia. Who wants to do the second? Who wants to do the G? Come on. We're gonna. I mean, you might as well get started doing this now when it's easy, because we're gonna do it in parasitology when it's not so easy, right? Yeah. Who wants to do that? Come on, somebody.
Burgundorferi.
Good. I like that.
I can't say it with my R's because it's English. So.
Yes. Well, I like bird. I like that. And I just go Dorferi. Yeah, bird Dorferi. Borrelia Dorferi. I thought it was a great name when I had to do a presentation on it. All right. So this one's Lyme disease. Where did it get its name? Are y'all familiar with Lyme disease? Maybe you should start there.
Doesn't it come from ticks?
Huh?
Doesn't it come from
It
ticks?
came from ticks. We just saw that with relapsing fever. We had
That's all I
a
know.
tick in the middle of that. Has anybody ever been to Lyme, Connecticut? That's where our first case was. So it spread from northeastern New England, spread across, okay? You got to the north, like Michigan, Wisconsin, the Great Lakes. Now it's just kind of just kept on spreading, right? At one time, Arkansas was considered a no Lyme disease state. I don't think we can say that anymore. Like one time we used to. We used to say, yeah, it's not here, don't worry about it. But it is. So it's an inflammatory disease leading to central nervous system, cardiac, and arthritis symptoms. So we have patients that have become debilitated with Lyme. Okay? It's led to these rare central nervous system and arthritis symptoms where they can't operate. They're at home. And it's debilitating. It is the hard body tick, this one. We had the soft one. So we have Ixodes scapularis, the hard body tick. And the specimens can be found skin, the blood, the cerebral spinal fluid. So this is our spiral key that's transferred through the tick bite. So Lyme disease, most commonly reported vector transmitted illness in the United States. 300,000 people yearly get flu-like symptoms, widespread complications. We get meningitis, we get facial paralysis. And since the black-legged tick is the primary vector, most prevention method is what? Keep the tick off, which is where you go out in the woods, put some deep woods off around your ankles, wear long pants, find the tick, get it off, okay? And then we have tick treatment. And what you'll find is most ER doctors, once they find a tick on somebody, they just go ahead and give them this antibiotic and say, should be good. But if your fever keeps coming and you get sick, come back and see us. CDC research shows the risk of becoming infected with Lyme increases each day the tick is attached, so we need to get the tick off. Check yourself for ticks. Remove the tick within 24 hours could greatly reduce your risk of Lyme disease. Mayo Clinic has also found a new species of bacteria causing Lyme disease in the Midwest. And this is where Arkansas got a pass. They said, hey, it's not Borrelia burgdorferi, that's Lyme disease, right? But what it was a very similar Borrelia mayomini. Is that how you say double I's? I don't know. Double I's, how do you say it? Y'all are not helping me with my English. It was found in samples from Minnesota, North Dakota, and Wisconsin. So we're kind of getting a blur of where we can find. And that's what they were saying. They were saying there was a tick-borne illness similar to Lyme in Arkansas that was getting confused with Lyme. And the diagnosis was saying it was Lyme disease, but this was a long time ago, right? I had this doctor from UAMS came and talked to us, and we were like, oh, you know, there's no Lyme disease. We hadn't had a confirmed test of Lyme. Because what they want to do is they want to confirm it in the tick. So if you take a tick off, put it in a little Ziploc, because if you go in, they're going to say, can we check the tick for the Borrelia burgdorferi, right? They're going to do a direct exam on the tick guts and see if they can find it. There was a new germ identified and described from the traditional Borrelia burgdorferi, and we used polymerase chain reaction to scrutinize the DNA of the bacteria. So these are bacteria, don't forget that. Now, the spirochete doesn't always sound like a bacteria, but it is. But we've come a long way with what? Polymerase chain genetic testing, and we can tell the difference between, and that's why I'm saying if you could keep your tick, and that way we can look at it and see. All right, so here's the soft body tick. This is the hard one. This is the one we mostly find on us, the ixoides scapularis. Does anybody like to tick hunt, or you want to go tick hunting? I have a great lab we could do if we want to go tick hunting. It's getting about tick time. I mean, it's already tick time, right? Yeah, you take a white pillowcase, clip on a string on each corner of it, and then you just go through the woods and you drag it behind you. And when you get through walking through the grass, then you look at your pillowcase and see how many ticks you picked up walking through whatever field you were in. It's a great way to find ticks.
What do you do with them afterwards?
You collect them and you go get a dissecting microscope if you're in parasitology class, and we open them up and look at them, right? Just reliving one of my college classes, huh?
That makes more sense than.
So we can do a direct examination. That's what we were getting at here, where we actually do a stain the smear. Diagnosing, unlike relapsing fever, we also have a serological test for Lyme. So we can actually check your serum for it, okay? Check your serum for antibody against it. Now this is key, and we had some students last year that said, hey, you didn't really, we missed this question. So I can go ahead and tell you there's a question on your exam associated with this statement right here, okay? Lyme disease, when the tick bites you, okay, you're going to get a classic erythema migraine pattern to the rash, which looks like a bullseye. We got a picture to show you. But the bullseye rash is known as erythema migraine. Okay, and they said I forgot to tell them that during lecture. I missed it on the test. I'm pretty sure this same slide they had last year. Pretty sure it had red writing here with bullseye rash. But when I asked about this was not in red for the answer. But the question said, the classic bullseye rash is known as erythema migraine. Okay? We have meningitis, we have cardiac problems, and we have arthritis. And what we're going to see is the reservoir for the Lyme life cycle, if you want to call it that, involves a white-footed mouse, a little field mouse, deer, birds can also be involved, but usually the mouse and the deer. Okay, so the tick starts out on the mouse, has a little larva, and then it moves to the deer. The deer runs all through the woods. These ticks fall off the deer. Life cycle's going good, and the reservoir host. So the deer and the mouse aren't affected by Lyme. The human is. And we know if you have a dog or a cat, the vet will tell you that it does affect your dog and cats too. So this is getting close to it, but that is the classic bullseye rash. Okay. So it was really fun. It wasn't really funny, but the last conference we went to, somebody showed up. One of our board members at ACLS showed up with a bullseye wrap. She loved going camping. And we're like, oh my gosh, you got Lyme. I mean, it was just like that, just beautiful red thing. And we, you know, we're all like zoomed in, like, you got Lyme disease. It's obvious. She didn't.
How did we not, or she not know that?
Well, she was just it just showed up that day. Like she'd been camping the weekend before the meeting started. And she doesn't remember getting a tick bite, but this showed up and it was the classic migraine there. She tested negative. She didn't have Lyme, but we were all just really, would have said 100%, yeah. There's a little white-footed mouse. There's the deer. Okay, so anywhere those two are together, Borrelia is hanging out. Borrelia is living in those reservoir hosts. And the tick is going to be the vector that transfers it off that reservoir host to you, the definitive host. And you end up with Lyme. So here we got the Lyme and Tick-Borne Disease Center in Columbia University, where a new organism has been discovered every 1 or 2 years with ticks. Okay, so ticks are not, they all have, could have a, you know, transfer of some illness to you. And we know, I mean, we'll go over this, but we know it's Rocky Mountain spotted. We know there's auriculosis. We know there's other tick-borne, you know, transfers. So it's just some history about how it all kind of got, you know, started going, what people think. And that finishes up our spirochetes. So our main two, we had syphilis and we have Lyme disease. Looking at those, I want to scratch my nose, but I'm blocked by my dream. And then that moves us to Legionella. Okay, Legionella is not a spirochete. I know it's really, to have it in this part of the lecture is confusing, but it's a pleomorphic gram-negative rod. So it could, you know, what's pleomorphic? Pleomorphic. Morphology, pleo, many different shapes, maybe. Maybe? Is that where we're going? Maybe. Gram-negative rod. We've already seen a gram-negative rod, right? We saw it on our gram stain. Right? We saw it ferment lactose on our McConkey. Who was our gram-negative rod? Remember? Gram stain. E. coli. Who said E. coli? Yes, E. coli. All right. So here we got Legionella. I know you hadn't heard of Legionella. We're not going to spend much time on Legionella, but it is a good one because we're still having what? We still have outbreaks of Legionella. We do. So the question is, has anybody been to Graceland Hotel and stayed there? No? Good, because when it was first built, the water supply for the air conditioner system was contaminated with Legionella. So this is still happening. There was an outbreak this month somewhere. I tried to look it up. We'll do it maybe. I'll look it up at lunch or the minutes before we start first lab. But there constantly, it seems like a more, you know, this is not one of those, oh, it started way back when they had a convention and hadn't heard of it since. There's still outbreaks with Legionella. Characteristically, catalase positive. So we know catalase, right? Who else is catalase positive? Staph. Thank you. Motility positive, and then gelatin positive, and we'll just leave that there for now. Non-spore forming, brown pigment. But what we want to look at is Legionella pneumophilia. Most commonly isolated species. Does anybody have a clue as to where it got its name? Well, if it's in contaminated water, and that water is being chilled for air conditioning, and you have a hotel you got to go all the way back here. Up. Maybe I can go back. I thought I had the article, sorry. But it was, anybody got their phone? We have a couple minutes and we'll finish up. Pick up your phone and look up me a Legionnaires convention in Philadelphia. I want to say it's the American Legion convention in Philadelphia.
In 1976.
1976, yes. Continue.
It just says the 1976 Legionnaires disease outbreak occurring in the Lake Summer in Philadelphia, Pennsylvania, United States was the first occasion in which a cluster of particular types of pneumonia cases were determined by the Legionella outbreak.
Yes, correct. I think it was the American Legion convention. But 19 what?
1976.
Okay, now look up Legionnaires at Graceland Hotel. See what year that was. I can learn a lot.
It looks, the news articles are from 2017.
2017, that's not too far ago. Right? Okay, so Legionnaires, respiratory secretions get in the lung tissue. There's a avoided production of aerosol. We do it by direct fluorescent antibody testing. We can check for this. But the key here, what I want you to remember with this, is we use a buffered charcoal yeast extract, which is a real dark media, like charcoal media. But that's definitely associated. We're going to associate that with some other things. But for Legionella, that's what we need to culture it. It's an obligate aerobe, okay, which means it's strict aerobe, but can grow an enhanced increased CO2. Growth rate 3 to 5 days. We get a convex gray glistening colony, and we detect it with indirect fluorescent antibody testing for serological testing. These are some old, there's the buffered yeast charcoal extract. Right, instead of, it's a black, 6 days of growth, ultraviolet light, lung issues, direct fluorescent antibody. Most healthy people do not get Legionnaire's disease. People over 50 years of age, risk factors, former smoker. They has the Legionnaire pneumophilia infection, where we get pneumonic legionellosis, acute pneumonia. But we also get a Pontiac fever, which is a non-pneumatic febrile illness. And most patients do not require antibiotics. All right. See you in lab this afternoon.